Professor Plomin wrote to me yesterday, worried that on a Friday evening, just before a coronation weekend, there would be no audience. But his name and fame have clearly drawn you all in. And there's a wonderful range of age in the audience too, with many undergraduates as well as senior members of the department and people from other departments. Third-year students will be very familiar with Professor Plomin's textbook, which is the definitive textbook on behavioural genetics. And many PBS students will have read, before they came to Cambridge even, Blueprint, Professor Plomin's popular work on behavioural genetics. He is professor at King's College London, and it's a great pleasure to have you speak.
Well, thank you, John. Well, thank you all for coming. I'm really very pleased to see so many people. Today, well, the DNA revolution has really transformed genetic research in all the life sciences, including psychology. And so what I'd like to do today is to tell you a bit about the DNA revolution, and specifically its findings in relation to what I call the genetic architecture of psychopathology. And the two worlds of genetics are quantitative genetics, like twin and adoption studies, and molecular genetics, DNA studies. And these two worlds of genetics went their own way for most of last century, but they've come together now. And I describe this journey in this paper. And what I'd like to do now is to focus on the last couple of decades, sort of like where the DNA revolution began. And it began with the fusion of quantitative genetics and molecular genetics. And the focus of this revolution is genome-wide association studies, which are techniques that allow you to identify some of the many, many small effects of DNA differences that account for heritability of a trait. And I'll tell you about what that's about. And what I'm going to talk about today is going beyond that, how that helps us with an area I call behavioral genomics. So there are three advances that made this possible. I know probably many of you know about genome-wide association studies, but it's the basis for material for what I'll be using in these further analyses. So in case you don't, I'll describe three technological advances that made this possible. The first was sequencing. I'm sure you've all heard about the Human Genome Project in 2003, published the sequence of most of the DNA bases, the 3 billion base pairs of DNA. And what was quickly discovered was that there is no human genome sequence. We all have millions of inherited DNA differences. And then people thought, well, maybe we could use those differences to begin to detect the DNA differences that contribute to the heritability of complex traits and disorders. But to do that, you need a lot of DNA, and that problem was solved by this other technological advance, PCR, which you all know about now from COVID, polymerase chain reaction. And what it does is it amplifies one little stretch of DNA and makes millions of copies of it so that you could detect minute traces of SARS virus that causes COVID, or detect differences between us in DNA. But it was still very difficult and expensive to do sequencing. And the thing that really made the difference was DNA arrays. DNA arrays are arrays the size of a postage stamp that can genotype hundreds of thousands of DNA differences throughout the genome very inexpensively, quickly, and accurately. Genotypes the most common type of, but simplest type of DNA difference called a single nucleotide base polymorphism, a SNP. And this is a stretch of DNA on one chromosome. The double helix is one chromosome. And this could be any two chromosomes, but they could be your two chromosomes. You inherit one of each pair from your mother, one from your father. And what we find is for over 99% of all the steps in the spiral staircase of DNA, they're the same for all of us. That's what makes us human. But somewhat less than 1% of those base pairs differ. And when it's a one base pair difference, that's what we call a SNP, a single nucleotide polymorphism. And that's why these chips are sometimes called SNP chips. So that's what I'll be talking about, are these inherited DNA differences called SNPs. Because it allowed you to genotype hundreds of thousands of SNPs, people realized, well, you could look at SNPs strategically located throughout the genome so that they tag most of the whole genome. It would be nice if you could just sequence all the DNA, which increasingly we're able to do because the costs have gone down so much. But the revolution was based on these SNP chips that tag most of the genome. And this is the way genome-wide association studies are reported. So what you do is you have to genotype a lot of people, and then you can look at a trait, say like this schizophrenia, and you could compare cases and controls and see if any of those SNPs differ. It's really just an association between that polymorphism, that variant, whether you have one allele or the other, and the trait you're interested in. And so the way it's reported is the x-axis here are these hundreds of thousands of SNPs organized according to chromosomes, so the 23 pairs of chromosomes. The y-axis is a p-value. I mean, it's like an effect size in a way. And this line is our p, typical p of 0.05 with 6 zeros in front of it because it's correcting for 1 million tests. It's a huge multiple testing problem. Because with GWAS, it really is, you're just looking at the correlation between each SNP and the trait. So it's a huge multiple testing problem. And above the red line then are SNPs that are significantly associated, corrected for this multiple testing. Each dot is one SNP. And the reason you get these peaks, this becomes important later, is because SNPs close together on a chromosome are inherited as a block. That's known as linkage disequilibrium. About each chromosome per meiosis has one crossover called a recombination. But if two SNPs are close together, they get inherited together. They're not recombined. And so linkage disequilibrium implies that SNPs close together will be correlated. So if one is a true hit, you'd expect others nearby to also be correlated. So that's why you get these peaks. But there are now hundreds, probably thousands of GWAS studies throughout the life sciences. And the biggest finding was that for most complex traits and common disorders, we're not talking about one gene, certainly, or even a few genes, not even tens or hundreds, but thousands and tens of thousands of DNA differences of very small effect. So that's what you get here a little bit too. The largest effects were so much smaller than anyone ever thought, imagined even. The largest effects are way less than 1% of the variance. And the average effect size in, like here, talking about 0.02%, 0.0002, incredibly tiny effects. And as psychologists, you know that means you need huge samples to detect it. So the first GWAS study of schizophrenia had 3,000 cases and 3,000 controls. And at the time, that was just thought, monsters. I mean, imagine 3,000 schizophrenic patients that you get genotypes on. And they're all carefully diagnosed as well. But now we're talking about 70,000. And so the name of the game now is to get larger and larger studies to be able to detect smaller and smaller effects. Okay, so that's what GWAS is known for. But what's exciting in my area is that it led to, you could use these GWAS genotypes not just to identify SNPs associated with a trait, but rather to estimate, say, heritability and genetic correlations. And I call it quantitative genomics because it's like quantitative genetics. It's like twin studies. You're trying to estimate bottom line influence of genes, heritability, on the variance of a trait, and the genetic correlation has to do with the covariance between traits and the extent to which genetics influences that. So I'll talk about that in just a minute, but the other major advance is polygenic scores. And given that the effect sizes are so small, what do you do with them? If you're a molecular biologist and you want to work from genes to brain to behavior, and the biggest effects are like 0.02% of the variance, that's going to be tough. But what you can do, as a psychologist you would know, is you can add up those effects and create a composite. You add them up weighted by their effect size, and you create a composite. That's all a polygenic score is. It's really like just, the reason you have items on a test, a questionnaire, is that no one item is reliable enough. but you have a bunch of items, you aggregate them, and you get a composite score. And that's what you're doing here. You can add them weighted, say, by their factor loading. So it's the same thing here. And these two, what I call behavioral genomics is quantitative genomics plus polygenic scores. And this paper describes these advances in relation to the future of behavioral genomics. And what I'm going to do today in the interest of time is just focus on findings relevant to this topic of the genetic architecture of psychopathology. Now, the question I'm going to ask is, to what extent does the genetic structure or architecture of psychopathology correspond to our phenotypic, the standard classifications that we use? And as you all know, DSM and ICD govern our research. You diagnose people according to these categories, and really clinical work as well. So these are very influential, this structure of psychopathology. But I don't know if I get much flack on this. There we go. That if you think about how they were developed, they're really all derived historically. Hardly any of our psychiatric disorders were derived empirically, and they're based on symptoms rather than causes. Nonetheless, they're assumed to be etiologically distinct. So much so that bipolar and schizophrenia, for example, until DSM-5, couldn't be diagnosed in the same individual. Why? Because, they're completely different. You can't have both. So there's a strong assumption that these are etiologically distinct. So the question I want to ask is, are they? To what extent does this, phenotypic in genetics, we call that, the observed, the traditional classifications correspond to the genetic structure. And a lot of this involves the genetic correlation, and that's why I need to just kind of go into it briefly. In univariate analysis, genetic analysis, like a twin study, we compare identical and non-identical twins to estimate the extent to which the variance of a trait can be explained by genetic variance. And heritability, then, is the proportion of phenotypic variance that can be explained genetically. These are path diagrams, which aren't just pretty pictures. They really are kind of algebraic representations of data, a model. And hx is the square root of heritability. It's the standardized partial regression of the trait on the genotype. Now, in bivariate analysis, though, we can do something else. Rather than studying the variance of one trait and then looking at the variance of another trait, we can look at the covariance between traits and the extent to which that's influenced genetically. And bivariate heritability is the extent to which the covariance between two traits, phenotypic covariance, is mediated genetically. And the genetic mediation is the heritability, square root, the product of the heritability of one trait, heritability of another trait, and this thing called the genetic correlation. So we need to distinguish the genetic correlation from that chain of paths. This sounds esoteric, but it does become important later in the talk. And to realize that genetic correlation is a derived statistic. It literally refers to the extent to which genetic effects on trait X are correlated with genetic effects on trait Y, independent of the heritability of the two traits. So that the heritability of two traits could be high, but the genetic correlation could be 0. Conversely, the heritability of two traits could be low, but the genetic correlation could be 1. It's independent of heritability. So you can derive the genetic correlation in any genetic study because we know HX and HY, so that they cancel and we're left with this derived statistic of a genetic correlation. Okay, now in twin studies, that's been done for a long time. And in the early 1990s, Ken Kendler, a famous psychiatric geneticist, looked at the genetic correlation between generalized anxiety disorder, whatever it was called back then, and major depressive disorder, and found a genetic correlation of 1, meaning that the genetic effects on anxiety are the same as the genetic effects on depression. The two are genetically correlated 1. So that means if you find the genes for 1, they're going to also be genes for the other. And we also looked at ADHD and ASD symptoms and found a very high genetic correlation between them. So most of the genes that affect ADHD, you might expect to affect ASD, which is kind of weird. We think of depression and anxiety as quite similar phenotypically in some ways, but we don't often think about a strong genetic relationship between ADHD and ASD. But this approach requires, you can only do this for common disorders where one twin can have one thing and the other twin the other. And even though I've always thought that was an amazing finding, and it's been replicated many times, it didn't take off at all. And that's just where it was left until the DNA revolution. Behavioral genomics provides several new general ways of getting at these genetic correlations using DNA directly. So I'll talk about quantitative genomics in just a minute, but first I'll mention polygenic scores because historically they came across first. The first GWAS study of schizophrenia was published in 2009. That was a study with 3,000 cases that I mentioned. And it was also the first study to publish a polygenic score because they also realized they were getting those hits I showed you earlier, many hits of small effect. And so our student, Sean Purcell, said, well, you could just add those up, weighted by the heritage. And so they did that, and they found they could predict 3% of the liability to schizophrenia. Now we're up to about 8%. Well, the most surprising aspect of that study was that they showed that this polygenic score for schizophrenia significantly it predicted bipolar, less than it does schizophrenia, but still substantially. So that, as I said, was really shocking because this is the first step in the nosology of psychiatric disorders. It's the first division, and they're thought to be distinct. But you can't estimate a genetic correlation from polygenic scores because they're tethered to the phenotypic variance. Just take my word for that. So what's been important is quantitative genomics has come up with 3 ways to use GWAS genotypes to estimate genetic correlations. And these are a bit technical, but they're very cool, and they're being widely used throughout the life sciences. So I thought I'd try to give you the gist of them, but I can't give you the details of them, but I give you the references for them. So bear with me on this. The first one is genome-wide complex trait analysis. So this was first published in 2011, and this was the first new way to estimate heritability and genetic correlations since the first twin studies 100 years earlier. So it uses GWAS genotypes, for hundreds of thousands of SNPs for each person in very large samples, thousands of individuals. And instead of like a twin study where you compare 2 groups, identical and non-identical twins, who differ massively, 100% and 50% in terms of genetic similarity, what you can do instead is take thousands of individuals, say 3,000 individuals, and look pair by pair for their overall genomic similarity. Like here's a pair that over all these hundreds of thousands of SNPs are a little bit more similar than other people. And then you say, does that difference in similarity, relate to differences in their phenotype? Now we're only talking about plus or minus 2%, but if you have 3,000 individuals and you look at them pair by pair, that's 4.5 million comparisons. So that's where the power comes from. So instead of just 2 groups, you've got 4.5 million groups. And it works. So you can estimate heritability. It's called SNP heritability, though, because it's reliant on the SNPs on the chip. And the chips have common SNPs on them, even though most SNPs are not common. By common, we mean certainly more than 1%, and most of them are greater than 5%, because you get more information from that in a GWAS study. But most of the SNPs, the vast majority of SNPs, are much rarer than that. So we've got a limited index of genetics here. But you can also estimate genetic correlation between traits. And it's the same idea. Instead of getting the covariance of the variance of one trait, in looking at pair similarity on height or whatever, you can look at the cross correlation between traits. So you can look at a member of a pair's height and the other's weight. And if there's a genetic correlation between them, you'd expect them to be slightly more similar than average. So it's the genetic analysis of covariates. And the really important thing about this is GCTA and these other quantitative genomic methods allow you to do this when you don't even measure the same trait in both people. So you don't have to find people who are schizophrenic and bipolar. And yet, just behind that is, if there's a genetic correlation between schizophrenia and bipolar, you'd expect if one person has schizophrenia and the other has bipolar in this very large matrix of unrelated individuals, then you'd expect that pair to be slightly more similar than pairs that where one's schizophrenic, where they don't, where they're not correlated, I guess I'll say. But the important point is you can do this for traits that can't be assessed in the same person, and it's really important for psychiatry. So the first application of GCTA was this paper in 2013. And people almost didn't believe it, but the genetic correlation they found using this technique between schizophrenia and bipolar was 0.68 for two disorders that are thought to be completely independent and distinct etiologically. They also found all three of the major psychotic disorders, schizophrenia, major depressive disorder, and bipolar, are genetically correlated. And they also found genetic correlations with ADHD and autism, ASD, autistic spectrum disorder, and ADHD. And even more magical is a second technique. So GCTA requires that you have individual genotypes for thousands of people. So that's hundreds of thousands of SNPs for thousands of people, and you're comparing them pair by pair. So you can see it's computationally intense. This other method, linkage disequilibrium score regression, LD score regression, is even more magical because it just uses summary statistics, summary effect sizes for the 100,000 SNPs across the whole sample. And the really cool part about this is you can do this from published data. If you publish a GWAS, you have to publish the summary statistics, the effect sizes of all the SNPs. So if you have the effect sizes, say, of the SNPs for schizophrenia, you can use LD score regression to get the genetic correlation with anything else, like depression. And this was the paper that described the method. And Ben Neal is one of our PhD students, I'm pleased to say. And in that paper, they published genetic correlations across scores, hundreds probably, of disorders and traits, because you can do it from published data. They didn't have to ask each investigator for the individual genotypes with which GDPR, is it GDPR? It's such a bane of my existence that I can't even talk about it. You see, it makes it very difficult to share individual genotyping data because supposedly you can identify individuals with that. So anyway, they published all those genetic correlations, and I pulled out from the genetic correlations, their correlations, something went screwy here, but between those same disorders. And these LD score regression correlations you can see, that is schizophrenia and bipolar and manic depression, are almost identical to those derived from GCTA. So I think that really made people believers. And since then, there have been a dozen or so papers that do similar sorts of things. This paper, 2018, in the meanwhile, we've had much larger studies, so you get more power, GWAS studies, and on more different traits. So this was a study not of 5 disorders, but of 10. And they found the usual sort of results, strong genetic correlations between 3 major psychotic disorders, as well as a number of other correlations in the off-diagonal. But one thing you can see is the matrix, the off-diagonal matrix is all positive. There's a positive manifold. Everything is correlated genetically with other things. There's only one negative correlation there that's just a very tiny, non-significant correlation. But what was important about this paper is that they did the same thing with neurological disorders. So they looked at 2, 4, 6, 8, 10, 12. They looked at 12 disorders, some of which are subtypes. And they found a very different pattern of results. The only significant correlations are within a disorder. So within epilepsy, there's two forms they talk about. They're genetically correlated. Within stroke and within migraine. So there isn't this kind of positive manifold genetic correlations between disorders. It's only within disorders. So something's peculiar going on with psychopathology. It isn't just an artifact of the method. The third method, and this is the last one, genomic structural equation modeling, is something you'll hear a lot more about now. It's genomic SEM. SEM is what's used to analyze twin studies. It's that path analytic model fitting structural equation model sorts of approach. And it's applied, this LD score regression applies it to LD score correlation. So it fits a model to that matrix of LD score regressions, which gives you a better description of the data, but it also tests the model rather than just giving you a narrative description. So it's a structural equation model fitting analysis of genetic correlations from LD score regression. And this has been used in several studies now, but I'll talk about the study that came out in the summer of last year by these same authors, Rotzinger, Michel Navard in Amsterdam. They present correlations for 11 psychiatric disorders now, genetic correlations from LD score regression. And they include some new ones like anxiety, generalized anxiety disorder. And that shows the largest genetic correlation in the matrix. And I hope that's familiar because that was the early twin study genetic correlation. It also shows the typical finding of a very high genetic correlation between schizophrenia and bipolar, and then also ADHD and autism. But two other new traits are anorexia and OCD. These are new GWASs that were done a couple of years before this. And they also show a strong genetic correlation. Now I'll talk about these other genetic correlations, but for now, I want to just note that this suggests a structure that's quite different from our current structure, and most notably that there's a psychotic cluster of schizophrenia and bipolar that are very highly genetically correlated. There's also internalizing, not so surprising, but it's still saying that these are very highly genetically correlated, major depressive disorder and anxiety, and a compulsive cluster with anorexia and OCD, and then a developmental cluster with autism and ADHD. Now it's a bit more complicated because one of the other new traits that were disorders that were a large GWAS was done for was PTSD. And PTSD complicates the picture because it correlates both with externalizing problems like ADHD and then also with internalizing problems like major depressive disorder and generalized anxiety disorder. So that's weird. And then the other two new GWASs are, so PTSD complicates this picture a bit. And then two other new GWASs are, where is that? Alcoholism, which doesn't really correlate much with anything else, and Tourette's syndrome, which doesn't correlate much with anything else. So they're not part of this picture. And genomic structural equation modeling, I know this looks a bit awful, but it's actually pretty straightforward. It's like a hierarchical factor analysis sort of thing, and it shows that the highest loadings on the first factor are anorexia and OCD, which we could call compulsive disorders. And then the next is psychotic disorders, which is schizophrenia and bipolar. You see these loadings are on schizophrenia and bipolar. And neurodevelopmental disorders, ADHD, autism, but also weirdly, PTSD. PTSD is also, as we showed, correlated with internalizing disorders, which is manic depression and anxiety. So these are very high loadings, and PTSD has a modest loading on that. So really what genomic SEM has done is instead of just giving a narrative description of the genetic correlation matrix, it tests a particular model. And in this case, this hierarchical model is the best fitting model. But what it also shows is a very important way in which the genetic architecture of psychopathology is quite different from what our traditional classification system. And that is that all of these factors are genetically correlated with one another. And that's a consistent finding in the literature, and it's called P, general psychopathology factor, analogous to G, which is the general factor of cognitive abilities. So that's a very important finding about our current classification systems, is that everything correlates with everything. That merely reflects the general the positive manifold I showed you in those correlation matrices. Everything correlates with everything. And we published a paper focused on polygenic P using 4 different methods: family methods like twin studies, GCTA, LD score regression, and GPS, polygenic scores. I didn't mention, I'm the only alone in using the phrase GPS, genome-wide polygenic scores, to distinguish polygenic scores created from scores. Hello, Claire. Come on in, make yourself comfortable. So GPS, to distinguish polygenic scores that come from SNPs across the genome from some polygenic scores that are just a handful of SNPs. And also, I can't resist a good acronym, so GPS is great because global positioning system, and it's a way of triangulating on genomic prediction. So in this paper then, we created genetic correlations for traits available for those data, and we found that family data give you a stronger genetic correlation matrix than, say, GPS. GPS, I told you, though, isn't really getting at genetic correlations. It's sort of discounted by the extent to which the polygenic score predicts the phenotypic variance, which isn't much. So the reason this is so powerful is we use the amazing Swedish registers of 3 million families. And so we actually used quantitative genetic approaches to estimate genetic correlations, primarily from full SIB and half SIB data, but also twins and adoptees. And you can see there's very strong genetic correlations among all those disorders. The weakest, and it counts, a first principal component accounts for 57% of the variance in that matrix. The weakest result was for polygenic scores, as you can see here, where first principal component significantly accounts for 22% of the variance, which is still a lot. And it's an underestimate, certainly. And GCTA and LD score regression are in the middle, 43% and 35%. So what this is saying is that not only is the genetic structure different at the level of those clusters, there's also this exciting finding of P, and there's lots and lots of papers now about that. And so the question I started with is, to what extent does the genetic structure of psychopathology correspond to the traditional diagnostic classification schemes that we use? And despite the fact that these studies are based on those traditional diagnoses, which assume independence, we're finding that genetic overlap is very much more the rule than the exception, suggesting that the genetic structure is quite different from the phenotypic structure. The second type of data involves, it follows up on this paper I wrote with the title, Common Disorders Are Quantitative Traits. It's the idea that we're not dealing with disorders. There aren't ideologically distinct classifications. They're quantitative traits from a genetic perspective. And this is easier to understand, so I'll go through it more quickly. I did just want to say, though, because clinicians often hit me with this, it's not to denigrate the extremes. There are problems. People definitely have problems. And clinicians have to identify those problems. That's true. But the point is that I don't think there's anything to be gained, and there's much to be lost scientifically, by reifying these classifications as ideologically distinct disorders. The simplest evidence, it's almost so obvious, but it's easy to overlook, and that is that polygenic scores derived from these case control GWAS studies are all perfectly normally distributed. That is, there's no breakpoint here for schizophrenia or bipolar or major depressive disorder. There's no threshold at which you suddenly you get diagnosed with that disorder. Now, this necessarily follows from the central limit theorem of probability because you've got a whole bunch of SNPs, thousands of SNPs, and you're putting them together. So that's what the central limit theorem is about. But as polygenic scores are used to predict genetic risk for psychopathology, it'll be impossible to ignore the fact that genetic risk is continuous. So one way, disorders are dimensions, which is another difference between the genetic structure and the traditional diagnostic system. One piece of evidence is that polygenic scores are normally distributed. And the second, and this is the last, so you only have to hang on to one more here, disorders and dimensions are highly correlated genetically. So even with quantitative genetics, we're able to get genetic correlations between disorders and dimensions using the same sort of approach I talked about, or between the extremes of a dimension and the rest of the distribution. And when you do this, you consistently find substantial genetic correlations between the extremes and the rest of the distribution. So there isn't anything genetically unique going on at the extreme. And we showed this with externalizing and internalizing behavior problems in children, with antisocial behavior in adolescents. The Swedish group published a genetic correlation about 0.9 between ADHD diagnoses and dimensions of ADHD, and autistic spectrum, as well as psychotic experiences. So everywhere we look, the genes that are responsible for the extremes of these distributions or disorders are the same as the genes that contribute to the rest of the distribution. And you can also do this with LD score regression. But can you see, if you follow what I said so far, you'll see that it requires that you have a GWAS for the dimension and a separate GWAS for the disorder, because then you could use those summary statistics and apply LD score regression to get the genetic correlation between them. So there's only two examples that meet this. The first was major depressive disorder and symptom dimensions. So there's separate GWASs for dimensions and disorders, and the genetic correlation is greater than 0.9. And the other example is with ADHD diagnoses and symptom dimensions. Again, the genetic correlation is greater than 0.9. So what I'm saying is there are no common disorders, just quantitative traits. Genetic risk isn't a matter of either or, qualitative, it's quantitative, more or less. Which means, in a way, that there aren't us normal people and those schizophrenics. We all have thousands of genetic risk factors for any of these disorders like schizophrenia. It's just quantitative, and the higher you are in this distribution, the more you are at risk for receiving the diagnosis. Okay, so the question I started with is the extent to which the genetic architecture corresponds to diagnosis. And the way I look at it, these are two kind of killer pieces of evidence that that our psychiatric classification system, it doesn't meet with the genetic reality. And how do we move forward? So in the last few minutes, and I do want to leave time for questions because I'm sure there'll be plenty. And the main roadblock right now is that all our GWAS are based on case control studies which come from traditional diagnoses. So it's hard to get out of that if all of your cases are diagnosed that way. So that circularity is the main problem. And what I'm going to suggest three ways in which we can move forward. One is something we can do now. It's not that great. And the other two are much more radical. And you have to be Pollyannish like me to think they might happen. But we need to recognize that current diagnostic nosology does not correspond to genetic reality. We're in a hole. We ought to stop digging and do something different. Not more GWASs on autistic spectrum disorder as traditionally diagnosed, especially when we've shown in I mustn't do this too much, I'm going over time, but in quantitative genetic studies, back then, the three components of autism, now two, are less genetically correlated than most psychiatric disorders are correlated with each other. And again, it's a good example. Why do we put those three things or two things together and call it autism? I don't think there's any answer other than Kanner in 1945 said they go together. He found some kids who had all three of those things. And sure, kids who have three of those things are worse off than kids that have two, worse off than kids that have one. But it doesn't make it ideologically the same thing. So why on earth do we do GWAS studies where your cases are autistic or not? Why not get it, the basic information? You get the information on the subtype things or the quantitative traits, you can always go the other way. But if all you get is a 1 and a 0, you can't go anywhere from there. So I'll suggest one thing we can do now. I think we can correct for P. And you can do that phenotypically, or you can take polygenic scores and correct them for the polygenic score for P, so that we're not counting that same genetic variance of P over and over again in every disorder. Right? So we can get at specificity that way. The best way, though, is GWAS by subtraction. So I do promise this is the absolute last method I'll mention, but it's a much better way of getting at this issue. We published this paper a couple of years ago, GWAS by subtraction. It involves a particular genomic SEM model as applied to LD score regression genetic correlation matrices. And the amazing thing about it is it allows you to do a GWAS analysis of an unmeasured latent trait. So if there's a trait that interests you and that for which there is no good GWAS by subtraction is one way that allows you to get at a GWAS of this unmeasured latent trait. So here's how it does it. It applies this model, this SEM model, and it is going to do a GWAS of a latent trait called non-cognitive that's independent of cognitive ability as they affect educational attainment. So here's the deal is we have good GWAS studies of cognitive ability and of educational attainment. In fact, the most powerful polygenic score in the behavioral sciences is for educational attainment because the samples are up to 3 million now. But we know that at least a third, closer to half of the variance of educational achievement is not due to cognitive ability. It's not just IQ. It's due to other things like motivation. Grit is really much to do with that. And also mental health, lots of other things. But we don't have good GWASs of those things. So this allows you to do a GWAS of this non-cognitive factor independent. And here's how it works. You do a GWAS, each SNP you correlate it with this latent variable. That latent variable, though, loads on both cognitive performance and educational attainment. So that's the SNP's correlation with the latent variable that subsumes both of those things. But then, you for that same SNP by SNP, for hundreds of thousands of SNPs, you look at its effect, its correlation with this latent variable, which is independent of that other one. So it's like it's asking about the residual effect. Once you take out everything due to those GWASs, can you predict and get a non-cognitive factor? And the answer is yes, you can. It gives you kind of interesting results. And the best reason for doing this is you can then create a polygenic score for non-P, a non-cognitive, so that everyone can use polygenic scores for these unmeasured latent variables. You do realize, right, that if you had a sample and you get GWAS genotypes, we routinely get 350 or so polygenic scores, including inflammatory stuff. There's so many interesting polygenic scores you can derive. And similarly, if you have a GWAS, you can then you can create these polygenic scores that predict these sort of latent variables. And this is what I think you probably twigged is what I'm doing now in my group for non-P. So the idea is we can do a GWAS, not just of each of these, but for P. This is how we do it. And we're looking for SNP correlations with a latent variable that subsumes all of these psychiatric disorders, including ADHD. But we can create a polygenic score for ADHD independent of P. And that allows us to create a non-P polygenic score that people can use. And so we'll do that for each disorder, independent of all the other disorders. Okay, but now that helps you get at specificity a bit, GWAS by subtraction, by correcting for P. But it still is circular, right? Because we're still starting with those diagnostic classifications. The two other approaches, I don't know if they're at all realistic, but they're certainly more radical, and that's to build a genetic structure from the ground up. And one way to do that, that's being done, is to use batteries of behavioral problems. So rather than starting with diagnostic classifications, just start with problems. And a good start has been made by the hierarchical taxonomy of psychopathology called HITOP. Is everybody familiar with this? Okay. Well, no. A good introduction is this recent paper by Ashwin and Caspi and colleagues on HITOP. But basically, it's a 5-level hierarchical model of psychopathology that starts not with DSM categories, but just with symptoms, problems that people have, maladaptive traits, huge numbers of them. And then empirically puts them together into what they call syndromes, and then those into higher order factors they call subfactors, and then higher order factors, spectra, which gets us into things like internalizing thought disorders. And then, because those are all correlated to some extent, a general factor of P. And this doesn't have much resemblance to DSM categories. It is just a phenotypic structure, though, but areas like personality and cognitive abilities that have all been built up forever from the ground up empirically, their genetic structure tends to correspond better to that phenotypic structure. The problem with psychopathology is it's not empirically derived, it's historically derived. Now, the other way is even more radical, and that is to think about behavior problems as the extremes of normal dimensions of personality. Because I'm already saying it is quantitative anyway. It isn't like you got some diagnostic, etiologically distinct disorder. And the nice thing about personality, it's also been psychometrically driven for a long time. Most people think about at least the higher levels of this as a 5-factor model with the acronym OCEAN. Probably all heard about this. Openness to experience, consciousness, OC, extroversion, agreeableness, neuroticism, and a general factor of personality has been proposed, although it's a bit controversial. And the nice thing about it is that personality traits are measured in normal distributions. If you've ever worked with symptoms, and this is HITOP's problem, they're J-shaped, right? It's a measure of how much of these symptoms you have. So it goes from 0 up. So it's J-shaped, like that, right? It's not normally distributed. So the low end doesn't really mean anything. But with personality traits, they're measured in normal distributions. So there's high ends and low ends. And so which is important because polygenic scores have low ends and high ends. Some people call polygenic scores genetic risk factors, but I really insist that's wrong. They predict the low end just as well as the high end. And what's nice about that is it'll draw attention to both ends of the distribution, as will personality traits. So for example, conscientiousness is considered a good personality trait. Kids do better, conscientious kids do better at school, for example. But what about the extremes? As Oscar Wilde said, most things in moderation. My mother always was telling me that because I was never very moderate. And I always liked Oscar Wilde's addition to that, including moderation. So at the extremes, you could think of the extremes of conscientiousnness as perfectionism, and the other end as impulsiveness or rashness. So I think this is something I'm going to think about anyway. I think it's a useful new way of approaching psychopathology. And it's interesting too, because if you think of autism, it's all focused on the high end, or eating disorders, say. What's at the low end? Who are these people at the low end? It isn't necessarily just low risk for that thing, like body weight even. What's a low polygenic score for body weight? I mentioned in my book, I show that my most extreme polygenic score is for weight. So this is the one near to my heart because I really do feel this genetic propensity to put on weight. But what's the low end? Is it just low risk for obesity, which is the way people think about it? It could also be problematic. I mean, maybe that's where you get into eating disorders, people who are finicky about their food. But nobody studies that low end. And so that's what I like about thinking in personality trait terms, that you can have a high end and a low end. So in summary, these are the data about genetic correlations in disorders and dimensions that lead me to conclude that the genetic architecture does not correspond to our psychiatric classification system. So what I'm suggesting, and again, it's probably Pollyannish, is that we need to move DSM to DNA. And I'm optimistic that in the next 10 years, the power that's coming, the energy that's coming from the fusion of these two worlds of genetics, I'm confident is going to help us move in that direction. Thank you very much.
Many thanks for leading us so clearly through such complex
Very
matters.
good audience on a late Friday afternoon.
Yeah.
There was nobody sleeping.
Nevertheless, we always have a break at this moment to let people who need to get away slip out, and then we'll have the questions. So if you need to get away at this moment, slip away now. Get away. Lots of people here that don't recognise psychology.
Okay, that's fine. So, John asked if I handle the questions, so please feel free to fire away. Want me to start? First question I get is, why genetics? Why focus on genetics? And that's what my book is about. I don't mention this because it's much more controversial, but I believe that inherited DNA differences are the major systematic force making us who we are as individuals. The environment's important. It accounts for about half the variance on average across psychological traits. But it's mostly, especially for psychopathology, what we call non-shared environment. It's not shared by two kids growing up in the families. It's not nurture. It's still environmental influences. But I developed the hypothesis that it's largely unsystematic. I'm writing a paper now on randomness and how we should embrace randomness. So, and what looks like systematic environmental effects are genetic effects in disguise. So when you find that parenting trait, parents who read a lot to kids have kids who read well at school, it's almost difficult to resist interpreting that correlation causally, because it seems so reasonable. Yeah, read a lot to your kids, you make them better at reading at school. But once you realize they share 50% of their genes, you can start thinking that it could be just a genetic correlation between them. And increasingly, and as a grandparent especially, I think parents are responding to genetic differences in their kids rather than environmentally causing those differences. So that's why I think it's important to focus on genetics. Okay, your turn.
Maybe I can just broaden that out a bit more,
Sure.
and ask you, how you think that gene expression and epigenetic effects relate.
Yeah, that's usually the second question I get in a public audience. And epigenetics, you're not really talking about inherited differences there. So when I say inherited, I mean inherited differences in DNA sequence. That doesn't change throughout life. You pick up some mutations, but it really doesn't change. And people say identical twins are different. They're really not. They're no more different from their twin than you are from yourself earlier in life, because you pick up a few random mutations. But you can do that. You can sequence identical twins. And for all of these SNPs, they're identical. So that's the inherited bit that's systematic. RNA is gene expression, and epigenetics is not counting alleles. It's counting number of transcripts of a particular gene that's being expressed. And RNA evolved to be responsive to the environment. Instead of just being static from the moment of conception, it changes all the time. As you're thinking, you're changing gene expression in your brain. So it's very labile and dynamic. And it's very important. Everything between genes and brain and behavior is important. But some people like epigenetics because they think the word comes from above genetics, and they think it's somehow almost like Lamarckian in a way. And those theories, there's a few examples of that in mice, but it's really not transmitted. There are two, Adrian Berg here, I think, shows that there are two major processes that wipe all these epigenetic marks when conception takes place. Because that's not what RNA does. It doesn't make you, it doesn't create stability across generations. What it's about is responding to the environment. The DNA does the former. That's what we inherit in the normally expected environment of the human species. So gene expression is important, but it really has little to do with what we're talking about. It's certainly not accounting for inherited differences. Are there epigeneticists here who want to argue? Okay, and I do think epigenetics is a bit past its sell-by date. You know, it's been around for a while, and it's hot, it's interesting, it's very difficult to do properly. DNA, you can get it from any cell in your body, because you started life with one cell. That DNA, half from your mother, half from your father, is the same DNA in every cell of the trillions of cells in your body. In contrast, RNA expression differs in all cells, in all cell types. It's state-dependent, age-dependent, and tissue-dependent. And you know, of course, what we'd like is tissue from up here, and other than from suicides, you don't get that. You couldn't even get undergraduates to agree to give brain samples for this work. So that's sort of my short take on it. But the main message is when we say genetic, we mean inherited DNA differences. Yes, sir.
This is probably not the third question typically asked. Maybe it's a stupid question, but I don't know much about genomics. But when you calculate a polygenomic score and you sum the effect size of a large number of SNPs, aren't you kind of assume that they have independent effects?
Exactly right.
Well, you mentioned that they're close together on the strand, but it seems that there's a lot of redundancy in systems. So you score high on some phenotypic dimension.
Yeah.
Is it likely you have a combination of strange polymorphisms across them?
Yeah, well, there's two parts of what you're asking. In the first part, as I talked about LD score regression and got past it, I realized I left out an important part. The reason it's called LD score regression, linkage disequilibrium score regression, is that it aggregates the summary effect sizes. You know, you don't need individuals. You can use summary effect sizes from a GWAS study published, but it takes into account linkage disequilibrium, because otherwise you'd be counting those correlated packages, blocks of SNPs several times over, and you wouldn't get an accurate estimate. So that's that. And I think the other part, you were moving towards like non-additive effects.
Well, even if the SNPs are far apart on different chromosomes, are unlikely to be affected by mechanics of mitosis, whatever it is, isn't it possible that to score highly on some particular phenotypic dimension, you probably need like unusual SNPs on a large number of SNPs, or it's given the system is likely to be redundant, lots of redundancy in the genetic to phenotypic mapping.
Yeah.
Isn't it likely that it's a non-linear mapping? So rather than adding up all your genes, it's like if you have this, and this combination, it's still particularly likely to have a high value.
Yeah, that's a very good point. And non-additivity just seems like it has to be important. In genetics, we talk about dominance is non-additive effects at a locus, and epistasis is dominance across different loci. It's non-additive effects across loci. What we're mostly studying are additive effects, the extent to which they add up. GWAS studies solely look at the additive effects of these genes. So if there was a lot of non-additive genetic variants, we wouldn't be able to detect these associations. But we can, and we know from quantitative genetic studies that most genetic effects are additive. And for example, the hallmark of a trait that is epistatic, it's like the idea, say if there was like a telephone number, you need 7 numbers to connect. A truly epistatic effect, if you had 6 of them, you don't get there. You need all 7 exactly right. So it's at the level of mechanism, that's certainly true. But at the level of the effect and the associations, they seem to be additive. And a hallmark is, if a trait is highly epistatic, you'd expect identical twins to be very similar because they have exactly the same genes. Whereas all other first-degree relatives shouldn't correlate much at all. And you don't find that much, except John here has found one for trichromatic vision, something, at a particular genetic level. But overall, the whole genome, the effects are additive, which is awfully good because otherwise we'd be out of luck in GWAS studies, because the power to detect these tiny additive effects, can you imagine if they're not additive? Just 2 genes, you multiply those, 3, triple, 4, so very soon you would not have a chance of detecting those effects. Yes?
I just would like to ask a very general social implication question from the Parkinson continuum. There's a huge move towards recognizing neurodiversity, and that's often on a particular spectrum. And I think what you're showing is that there's a lot of neurodiversity in all of us. We're all on multiple spectrums, where we're here on one and there on the other. But on the other hand, and some of them are not actually all that positive. They would have a lot of suffering associated with some of these dimensions where we normally distribute it on. On the other hand, there are some groups who really benefit from having a diagnostic label, and they want to put a cutoff. So there are communities of people who call themselves voice hearers and think and would like to see it maybe as a dimension, but other communities who would really enjoy or benefit, psychologically benefit from having particular labels. Just was wondering if you could speculate on the social implications of these continuum.
Well, I do get that a lot. Parents, I just had a friend who said, now I understand, my kid has ADHD. So that sort of explains everything. So it gives her comfort. But, as a scientist, that doesn't govern our science, right? If that's what you need to tell people to make them feel better, okay. But I don't know, I would rather know the truth. And as I talk to people with diagnoses, you say, these are all quantitative dimensions. And that's good, because you're not looking for a cure. If there's no disorder, there's no cure for the disorder. There's just ways of ameliorating it if you want to. So it's all quantitative. And I find when I talk to people about it, say people with schizophrenia about it, people do understand that. And in a way, it's kind of a relief. I know what you're saying. There's some comfort in saying, this is what I've got. But on the other hand, there's also comfort in knowing it's not like you're outside the human species. You're different at a basic level from other people. You're at the extreme of a dimension. And I think I'm just saying, I think you can talk, my experience is I can talk to people about that. But, there is that bit that you're saying. But I don't know if it's such a good thing, though, to give them wrong information.
I was just curious about what you make of the broad implications. So, for example, in this university, we want to assess students inclusively, right? We want everybody to benefit. But now, from what you say, it's not just one continuum. It's multiple continuum, and it's not just going on one side, it's two sides. So some people would benefit from the exact opposite of what would benefit the other continuum. So I'm sure you thought about this quite a lot.
Yeah. Do you have thoughts about that? That's, as far as I've gone thinking about it, is, that in general, I'd like people to tell me the truth. And I wouldn't like it if my doctor's telling me something because they think it's good for me, even though they know it's not true. And I do think there's ways of reconciling yourself to the idea that it's dimensions and these, we're at different points of all these things. It's not just a matter of are you normal or are you abnormal? And I think no one's done that yet with neurodiversity, which has been kind of tied into traditional classifications or into categorical systems. But I don't see why you can't just recognize neurodiversity at a quantitative level. So that's what I'd push for, but not something I know much about. Yes, sir.
I might have got this wrong, but I think you said at one point that for schizophrenia, a few years ago you could explain 6% of the variance, and now you could explain 9% of the variance, or something like that.
Yeah, the first one was 3%, and
okay.
The most recent one I think is like 7% of the liability.
How do you interpret that figure? Where's the rest of the variance? And is that going to continue? We're going to continue to explain more and more as the techniques improve, or is something not genetic? Or...
Yeah, in the cognitive realm, we have the best data here because you can get very large samples. So as I said, for educational attainment, which is a lousy variable, isn't it? It's just years of schooling. But every GWAS study, as demographic information, asks people about years of education. And so that's why the recent paper has 3 million people in it. And the idea is, you go from 2013 was the first paper that accounted for 3% of the variance, and it's been a pretty sharp rise up to now about 15% of the variance is explained with 3 million people. Educational attainment's a crummy variable, though. We can use that to predict more variance, say, of educational achievement, tested achievement, and we can explain close to 20% of the variance on GCSE scores. Now, the heritability of GCSE scores is 60%. So even there, we're still a long way from it. But in 10 years' time, as things go from 0 to 20, is huge. And the rest is called missing heritability. And if I could have one wish, it would be to understand what missing heritability is. It's beginning to look like these brute force approaches, by just getting bigger and bigger studies, they're asymptoting. You know, even with depression, a paper just submitted last week, it's explaining something like 6% of the variance, but it's doubled the sample size to something like 150,000 or something. So that brute force is only going to take us so far. But again, being an optimist, I think this isn't our problem. This is throughout all life sciences. So some of the brightest people around are working on these issues. So I'm optimistic that we'll go beyond just brute force approaches, smarter approaches. And with AI, I think, you know, you can think of, it's possible. I haven't asked GPT yet, but I think it will be possible to do that. But I would like to say, though, that even we can now explain 15% of the variance of GCSE scores. With the new polygenic score, we will be able to get close to 20%, which, and people say, well, that's only 15%. But you know, in psychology, we often don't look at how much variance we're explaining. You know, we're still stuck with statistical significance, which is really responsible for a lot of the replication crisis. And just think about things you know in psychology, and what's the biggest prediction you can make? Well, what about GCSE scores? I asked people at the Education Institute when I talked there, how much variance in GCSE scores can you explain by these expensive Ofsted school ratings? You know, 20,000 each, 2 days of examiners going in. They're very good assessments of school quality. So how much variance do they explain in GCSE scores? Well, they ought to explain a lot, because it changes people's lives, right? I know lots of people, they move across the street so they're in the right district, for example. They send their kids to the best schools for hundreds of thousands of pounds so that they will supposedly do better. Well, the answer is 4%. And kids aren't randomly assigned to schools. If you correct for SES, it's 1%. And yet we're making huge decisions in life based on that. It's not to say it's unimportant, and this is only related to school achievement, GCSE scores. And you know, that's sort of what people say they're doing, but we know that a lot of them want their kids to be with the right sort of kids, you know, that sort of thing. So it's important to put it in context, and explaining 15% of the variance is a lot. I just found another one example like that the other day. What about smoking and lung cancer? It's actually very hard to find these data. Maybe there's an epidemiologist here, but I asked George Davey-Smith, and he gave me the answer, but I'm still trying to track down the reference for it. But you know, 80%, 85% of the people who have lung cancer have smoked. But only 15% of people who are lifelong smokers get lung cancer. So the odds ratio is very high, but the variance explained is less than 10%. So if in the population you're trying to predict who's going to get lung cancer, and you measure how much someone's smoking, you'll explain less than 10% of lung cancers, you know, the 3 major types of lung cancer. So, you know, this 15% is really a big deal. And a lot of things we make a big deal of in psychology don't account for any variance, like gender differences in cognitive abilities. You know, boys are from Mars and girls are from Venus. Girls are good at verbal stuff, boys are good at math. It accounts for less than 1% of the variance. It's highly statistically significant because you have these huge samples. But the variance explained, the effect size, is very small. So I'll get off my soapbox now.
Can we come back to the idea of interactions? I thoroughly agree with you that we don't really want them because they're very hard to handle. So that our initial sort of attitude of them should be war. But sometimes we are forced to admit that they occur, and they can be very interesting at the level of explanation. Going back to your very nice structural equation model, your multi-level factor analysis, which is behavioural, would you think it worthwhile fitting interaction terms, difficult though that is in SEM, would you think it worthwhile to say, look guys, there really is only one interaction here, or there are two, or whatever? Would you say that that should be a part of the the scientific strategy to face up to the possibility of interactions in order perhaps to be able to say, well, you don't need to worry because there aren't really many strong ones.
Yeah, good question, Mark. But do you mean interactions between what? You don't mean at a single SNP level? Do you?
No. I mean that the factor, in terms of the observables, is there an interaction that you can detect between two of the intermediate factors in producing the variable at the next level down?
Well, as you say, it is empirical, and when people have tried to put in interaction terms, power kills you, but there's no great evidence for interactions. Now, I didn't, in that paper I have on the history of genetics, an important phase before GWAS studies was candidate gene studies. The 1990s, when it first became possible to sequence DNA, you could measure specific DNA differences, and yet it was very slow, very expensive. So what we did as psychologists, we looked at a few SNPs that we call them candidate genes, like serotonin and MAOA, and dopamine for ADHD, because that's part of Ritalin. And there were hundreds of studies reporting positive results, and that was genomics' contribution to the replication crisis. They just don't replicate. And you do everything wrong. You're looking at a lot of SNPs, a lot of traits, you pick the one that shows an interaction. But it got so bad that journals won't publish candidate gene environment interaction studies now unless you replicate it. So as reasonable as it is to look for interactions, most of the effects you get are additive. But it's empirical. If anyone looks at more complicated interaction models and predicts more variance, great. And that's what I'm in it for, is prediction. Anything that adds to the prediction is great. And with polygenic scores, I didn't mention, you keep adding SNPs as long as they add to the prediction in an independent sample. You're not just taking statistically significant results, because the effect sizes are so small that P of 5 to the minus 8 is ridiculous, because you need a sample so large to detect it, And it turns out, empirically, a lot of those other SNPs, way less than statistically significant, contribute to the prediction of a trait in an independent sample.
Could I ask a question as
Yes.
as a devil's advocate? The SNPs that go into a GWAS are just traditional markers. The mischief is usually done by mutations nearby, and you rely on.
No, the SNPs are mutations. So, all DNA variant starts as mutations.
Well, they're polymorphisms.
Hmm?
They're polymorphisms. I mean, they're all sort of 40, 60 in the population. That's why they're in GWAS chips.
No, actually, most SNPs are much less than 1% allele frequency. So they're very much rarer.
Yeah, but on a GWAS array, they're all common.
So, all SNPs are, 5% and up, so that's 5% versus 95%.
Yeah.
But the point now is to look at those that are much more rare, like 0.001, 1 in 1,000 sort of thing. But I'm just saying all DNA variation is a mutation. These SNPs are all mutations in single base pairs, initially. And some of them, if they have a negative effect, they get wiped out of a population over tens of thousands of years. So you're talking about de novo mutations that occur in one individual?
No, I'm talking about the last decade's GWAS, which use common SNPs that are polymorphisms, which may have been, many of these polymorphisms have been in the primate lineage for 1 million years.
Well, not 1 million, but they have arisen as a mutation initially, and some of them persist.
Yeah, and the population geneticists understand why polymorphisms are maintained.
But in the common ones are interesting because they've been probably around for a long time, because they start in one individual. And why do they spread through the population? They must have some advantage to reach a 40, 50% frequency.
Yeah.
But they all start as a single mutation in a single individual.
Yes, but now they're polymorphisms and they're the markers in GWAS. Anyway, let me go on. you know, I'm really just an honest visual scientist and learned my genetics from ophthalmological genetics in the 1990s.
Yeah.
And the ophthalmologists were successful with candidate gene studies for a particular reason. Inside the cones and the rods of the retina, there are about 50 proteins that are specific to rods and cones. And it's easy to harvest rods and cones, you know. And a common cause of blindness is retinitis pigmentosa. And in the 1980s, there were, say, 3 clinically distinguished types of it. Dominant, recessive, X-linked. The geneticists started with the common idea, well, rhodopsin is a major protein in the rods of the eye. We have this disease, retinitis pigmentosa, where the rods decay. Let's sequence rhodopsin. And about 30% of British cases had a mutation in rhodopsin. But no 2 families had the same mutation. And the geneticists then worked through the other proteins that are specific to the wonderful little world of the photoreceptors and soon could account for most cases of retinitis pigmentosa. However, in rhodopsin, there may be a lot of mutations that cause retinitis pigmentosa, but nearby them are mutations that cause a quite distinct condition, stationary night blindness, which clinicians would completely distinguish. Now, if you did a GWAS of retinitis pigmentosa, which no one has bothered to do, I think, because you don't need to know it, you would find associations with stationary night blindness and a particular SNP, and retinitis pigmentosa and a particular SNP. And the SNPs that are doing the mischief may be very close together, different mutations nearby. So could this model transfer to bipolar and schizophrenia by saying, yes, there are proteins that are central to the functioning of the central nervous system, but there are different mutations underlying schizophrenia and bipolar disorder in the same proteins. This is, I'm just being a devil's advocate.
No, I think that's, it's good, it clarifies some things. I didn't really emphasize that we're talking about common disorders here. And there are 10,000 rare single gene disorders. And they're operating much like you're talking about. There is a gene, and usually you find, like with phenylketonuria, which causes 1% of severe mental people in institutions for intellectual handicap. There are, I think, hundreds of mutations in that gene. Some of them have a bigger effect than others. And that's generally true. So the SNP will target a particular area, maybe the gene, maybe not the right regulatory area. So if that's the point you're making, yes, I agree completely that it's not necessarily the functional DNA variant. And then I think the other point you're making is it could be other genes nearby. It could be within the gene, it could be something in a regulatory region or not. We don't know the biological mechanism. It's going to be hard to figure it out, though, when the biggest effects account for so little variance. Because these are not like things you're talking about, which look like single gene traits. With a single gene disorder, you almost don't need to be a geneticist to see them because the transmission patterns across generations are so obvious. So I don't disagree, I think.
Well, we can continue later. You have a question there.
Time for a broad question. You've spoken a lot about the predictive ability of using genetic studies, specifically in terms of like diagnosis or dimensions, whichever way you want to categorize it. But I was wondering if you had any thoughts that you wanted to share about using it for things like precision psychiatry, so predicting the impact of treatments or the utility of genetics in treatment.
Well, so I don't know if you could hear the question. It's about whether or not you can look for genes, say, as they interact with treatments, and that's a huge area of research now. You can't do a large study of anything all the cohort studies, for example, collect DNA. So in depression, for example, there are these randomized control trials comparing the effects of drugs and CBT, for example. They all get DNA because what you want to see is maybe they don't work very much on average, but perhaps they work particularly well, say, with people with a strong genetic disposition. Maybe drugs work better for those people. Whereas you could also find maybe with genomic prediction, maybe some drugs are very bad for some people. That's actually driving a lot of it in the medical area. That with pharmacogenomics, that's kind of the biggest area now. Yeah, so I think there's a lot of interest in that.
Do you think there's any, when you talk about this revolution then, any space for a categorization of predicting people in terms of what they might react best to, rather than categorizing people into what they suffer with, rather categorizing them in terms of what they will respond to?
Yeah, but the GWAS studies are predictions of a disorder. But you could do a different type of GWAS along the study lines you're talking about, where you could actually do a GWAS study for interactions. And that's being done. It's a power issue. And I think there's a whole bit about personalized genomics, and there's a strong resistance to it, that people say, oh, it just doesn't predict enough. But as I was saying with the example with smoking, it actually does predict quite a bit. And people talk about stratified rather than personalized. Okay, maybe for an individual, you don't have a lot of prediction, but you could say for those people in the top 3 standard deviations above the mean, the relative risks are getting for schizophrenia onto something like 20. So it's a very substantial increase in risk at those extremes. But I still think of that as individual differences. It's just the extreme of this distribution. So, yeah, okay. Well,
I think it was a very good discussion. Are you able to stay a little while and have a drink with us? Or?
Yes.
Yes. So, by tradition, anyone who wants to continue the talking to Professor Plomin can join us in all bar 1 in Regent Street, and through the generosity of the psychology department, we will buy you a drink.
I'm afraid I can only stay for about half an hour.
Yeah,
Okay.
So we'll go briefly to all bar 1, and anyone who wants to continue the conversation can come there. And I'd like to thank Professor Plomin